Hey everybody, it's Miss Manili. Sorry I can't be here today. I'm at a meeting, but I thought I'd go ahead and give you a brief introduction to experimental design. Once we finish the video, I'm going to give you some time to work on your projects, and I'll talk about that in a little bit. So far in our second unit, we've talked about designing samples and things we have to be careful of to make sure that the samples that we design are done properly to reduce bias. The same can be said about experiments. There are some factors that we need to consider when we're designing experiments so that our data is worthy of further investigation and so that we can draw conclusions about causation among our variables. So just to start, some of the things that we're going to talk about in this particular activity, we've already talked a little bit about the difference between an observational study and an experiment. We're going to talk a little bit more about the concept of confounding, and then we're going to go through some definition and some terminology that relates to experiments. For example, the difference between units, the different types of variables, explanatory and response, which we talked about in the last unit, along with treatments, comparison, random assignment, control, and some other issues that relate to experiments and not to samples. So first of all, let's just kind of review what we talked about at the beginning of the unit, the difference between an observational study and an experiment. An observational study observes individuals and measures variables of interest, but it doesn't attempt to influence the responses. That doesn't mean that there's not an interaction between the investigator and the subject. So for example, the investigator may question the subjects or ask them to complete a questionnaire, or they could simply observe behavior in the natural environment. An observational study would include reviewing records. It could also include administering tests to gather information. And the test could be anything from like an IQ test, a personality inventory kind of test. It could also be, a lab test where they're drawing blood or perhaps doing a brain scan. But the purpose isn't to change the effects or to cause a reaction. It's simply to determine information that already exists. And there are different types of observational studies. I'm going to talk to you about two of the common types of observational studies. One is called a retroactive study, and it's basically, where we identify an outcome of interest that has already occurred, and then we kind of go back in time to figure out what perhaps may have led to the occurrence. So an example would be the incidence of autism. There's been a much greater incidence of autism over the last 20 years than prior to that. And so obviously, doctors and medical researchers want to know why. Why are more children being diagnosed with autism than ever before? And so when you look at the children with autism and go through their medical records or even factors in their environment, we're looking to see, what kinds of factors or what kinds of characteristics do these children have in common that may be different from children who do not have autism. And so an example would be, looking back to medical records and looking at vaccinations. There was a time when it was thought that vaccinations actually were the cause of autism, and that research has now been debunked, but there was quite a bit of questions about whether or not the vaccinations were the cause of autism. And because this is an observational study, it's very difficult to conclude causation. So there was a big kind of frantic upheaval when it came to thoughts of vaccinations causing autism. And you had a lot of parents that were concerned about giving their children vaccines because of it. Another type of observational study would be a prospective study, where researchers identify the subjects and then collect data as the behavior occurs or as events unfold. A longitudinal study is an example of this, where data is gathered for the same subjects repeatedly over a very long period of time. And longitudinal research projects can extend over years or even decades. An example would be studies that were done regarding the Head Start program, which I believe began in the 1970s, which is a preschool program for disadvantaged children. There's for families living in poverty to give those children, it was like a school readiness project. And so these children who began the Head Start program were followed over the years to see, how successful they were in elementary school, in middle school, and so forth, to see being a part of Head Start actually did help them in the long run prepare for schooling. So that would be an example of a longitudinal study. Both examples that I've shared are observational studies, and so it's very difficult, like I said, to conclude causation. So one of the things we're going to talk about in the design of experiments and the different designs of studies is to diagram them. And so the example you see right here is a diagram for a retroactive study. And you'll see that the study begins with the disease itself. So basically the outcome of interest has already occurred at the time the study is initiated, and then the study or the design allows the investigator to formulate ideas about the possible associations and investigate potential relationships. So an investigator conducting a retroactive study typically utilizes administrative databases, medical records, interviews with patients who have already been known to have the disease or condition. So example, again, would be the incidence of autism, looking at children with autism and reviewing records to see the differences, or looking, and at the same time, looking at children who do not have autism and looking for similarities that might explain the cause. And so when it's looking at the exposed versus unexposed, I think one of the things that they were looking at was vaccinations. And that's just one example. In a prospective study, you define the population in the beginning. So, for example, I might be looking at children living in poverty for the Head Start program, and then we have the sample is self-selected. Obviously it's going to involve some bias because either the researcher is choosing the subjects to participate or the subjects volunteer to participate. And then we look, or we compare children who were in the Head Start program versus the children who were not part of Head Start. And then over time we look to see how well they perform in school. You could also do this, for example, with smoking or substance abuse. You might have a group of college students, for example, and compare the college students who may drink frequently versus the college students who don't drink frequently, and then determine, observe them over a period of time to determine if any conditions have developed. Maybe compare grades, compare risky behavior, compare legal matters to see if there's a difference between the group of students who drink regularly versus the group of students that don't. And of course, there are other variables that are going to be at play here that, again, make it difficult to determine causation. So let's talk about that, correlation versus causation. We saw some slides in the last unit that kind of showed you some interesting relationships between variables that were completely unrelated, but yet had very strong correlations. And so while correlation between variables may be noted in an observational study, a causal relationship can't be determined because there may be other variables that contribute to the outcome. And confounding occurs when two variables are associated in such a way that their effects on the response variable can't be distinguished from the other. And when our goal is to understand cause and effect, it's important that we only do a well-designed experiment. It's the only source of truly convincing data. So, for example, if you think back to the longitudinal study, where is it? If we're thinking about like the Head Start programs, for example, there may be a lot of other factors involved with the children that end up in Head Start versus the children that don't. There may be a lot of differences among those group of children to begin with that might explain the differences in their outcomes. An experiment is different from an observational study. An experiment is where we deliberately impose what is called a treatment on the individuals to measure their response or reaction. And in a well-designed experiment, all of the other variables are controlled, so it can be concluded that any observed reaction or response is caused by the treatment alone. And the distinction between an observational study and an experiment is very important in statistics in terms of the conclusions that we can draw from the study in terms of inference. And we'll talk about that a little bit later as well. Sorry, the snoring that you hear in the background is my dog, who is sleeping soundly next to me as I record this. I'm going to go through some terminology in experiments, and this is going to be important for your quiz, obviously, and also when you're reading about research, you're going to see these words all the time. So it's important to know the terminology. An experiment is a statistical study in which we actually do something to people, animals, or objects. And we call these the experimental units. And we do it in order to observe a response. So the experimental units are the smallest collection of individuals to which the treatments are applied. Individuals could be rats, bacteria, they could be products on an assembly line, whatever. When the units are human beings, then we call them subjects or participants. And a specific condition applied to the units is called, in an experiment, is called a treatment. And again, it's just that condition that we impose to gauge a reaction. We often want to combine several explanatory variables. And when this happens, we often use a combination of what we call levels and factors in the experimental unit receives. The factors is basically one of the explanatory variables of an experiment. They mean the same thing. Factor is an explanatory variable. And there always has to be at least one factor in an experiment. Many experiments study the joint effects of several factors, and in such an experiment, each treatment is formed by combining a specific value or level of each factor. So, for example, if I'm studying the effects of a weight loss supplement, I might want to look at taking 500 milligrams of the supplement versus 200 milligrams of the supplement. Or maybe I want to explore the difference between taking the supplement twice a day versus taking the supplement once a day. Those would be different levels. Oops, I didn't mean to go. And then basically to determine the number of treatments, you're going to multiply the number of levels for each of the factors in an experiment. I might also, let me give you another example where there might be two explanatory variables. Let's say I'm looking at exercise and a supplement. So with exercise, I might be looking at individuals who exercise 30 minutes every single day. I might also want to look at exercising 30 minutes every other day. Or maybe I want to look at exercising 1 hour every other day. Those would be the levels of the factor of exercise. And then with the supplement, maybe I'm looking at different milligrams. So maybe I'm looking at 250 milligrams versus 500 milligrams. And those would be the different levels of the factor of the supplement. But in order to determine my treatment groups, I might look at individuals that will be taking the supplement at the 250 milligram level, combined with exercising for 30 minutes every day. But I also want to look at the 250 milligram level with participants that are going to be exercising 30 minutes every other day, and then also 1 hour every other day, and compare which combination is going to be most effective in reducing weight. So that would be what we mean by combining the treatments and multiplying the number of levels times the number of factors. And so here's an example. An ad for OptiGrow plant fertilizer claims that with this product, we will grow juicier, tastier tomatoes. We'd like to test this claim and also see if we can get by with half a dose. We will use 60 tomato plants of the same variety obtained by the local garden store. So let's see how well you can respond to these questions using what you know about the terms that we just talked about. Go ahead and press pause and answer these questions, and then when you're done, I'll go through the answers. Okay, so the answers in this case, the experimental units in this example are going to be just the 60 tomato plants from the local garden store. The response that we're interested in would be however we're going to measure the juiciness and the tastiness of the tomatoes, whether there's some sort of scientific test that we can use, or if it's simply just a matter of opinion and we have a survey of individuals who actually, bite into the tomatoes and then measure the juiciness and tastiness that way. So it's not clear how that's going to be measured, but that would certainly be the response that we're interested in. The factor in this case is going to be the plant fertilizer, the OptiGrow itself, and the levels would be the different doses. So we've got the full dose of the fertilizer along with half the dose. And again, we don't know what constitutes a full dose, but in any case, it would be the full dose and the half dose. Those would also be the treatments, because those are the only two options we're going to have. We might also want to consider a control, and we'll talk about that in a little bit as well. So the treatments then would correspond to the factors, either the full dose of the OptiGrow and then the half dose of the OptiGrow. All right, let's extend this situation a little bit further. The makers of OptiGrow want to ensure that their product will work under a wide variety of watering conditions. So let's include this as a second factor. Specifically, let's look at daily watering versus watering every other day. Now, how many factors are there, and what are they? And what are the levels of the factor? Now, what would be the treatments of the new experiment? And then you're also asked to draw a diagram. I'm going to give you a chance to actually just go ahead and construct the diagram using your own thinking. Like, how would you draw this out to somebody so that they would understand how the plants are going to be assigned to the treatments and what those treatments are going to look like? Please press pause, and then we'll go over the answers in just a second. Okay. So now that we've added the watering conditions to our experiment, we have two factors. The OptiGrow fertilizer is one of them, and then the watering is the other. What are the levels of the factor? Well, we've already discussed the levels for the fertilizer. We had the full dose and the half dose. But there's also two levels to the watering as well, and that would be watering daily versus watering every other day. And so now if you think about it, there's going to be several different combinations of those factors and levels. And so sometimes it helps us to draw a picture or draw a table to kind of sketch things out. So now that I have two different levels for my OptiGrow, I've got the half dose as well as the full dose. I've also got my watering every day versus every other day. I've got 60 plants, so that leaves me with 4 different treatment groups. 15 of those plants, I'm going to end up assigning them randomly, are going to be watered every day and be given half the dose of OptiGrow. Another 15 are going to be watered every other day and be given half the dose of OptiGrow. I've got 15 plants that are going to be watered daily with a full dose, and then 15 that are going to be watered every other day with a full dose. And then what I'll do at the end of my experiment is compare the juiciness and the tastiness of the tomatoes under each of those conditions to see which combination of levels and factors produced the best tomatoes. Here's another example. Many students regularly consume caffeine to help them stay alert. So it seems plausible that taking caffeine might increase an individual's pulse rate. Well, is that true? One way to investigate this claim is to ask volunteers to measure their pulse rates, drink some soda with caffeine, measure their pulse rates again after 10 minutes, and then calculate the increase in the pulse rate. Suppose the pulse rate of every student went up. Can we then conclude that the increase in pulse rate was due to the caffeine? And then you're asked to explain your answer. Well, go ahead and press pause and think about this for a second. Even if it were true that the pulse rate of all of the students increased over that 10-minute time, are we convinced that it was because of the soda? And one of the things we have to think about is, were there any other variables involved? You know, for example, did some of the students do jumping jacks to kind of stay awake, or were they more alert because they had to take their pulse rate? Did that sort of wake them up and then suddenly increase their pulse rate? So there may be other variables that are at play that may explain the increase in pulse rate, and we're not sure just by looking at the given information. So one of the things that you have to be careful of, and this would be how to experiment badly. This would be a very oversimplified way to take an experiment, where you have your units, you apply a treatment, and then you measure the response, like we did in the pulse rate, like drinking caffeine and then seeing if there's a difference. In the lab environment, simple designs can work because there's nothing else affecting. Like if you're dealing with cells in a petri dish and you add alcohol to them or you add sugar to them and there's a change, because they're in a petri dish, you know that there aren't any other variables impacting that. And so you've eliminated all the other variables and you can kind of associate any kind of response with the single treatment. But in the real world, if I'm dealing with, especially with human beings, there may be other things at factor or other things at play when considering a response. So in field experiments or experiments with animals or people, we deal with more variable conditions. And outside the lab, badly designed experiments often yield to worthless results because of this issue called confounding. A well-designed experiment includes measures to control these other variables. So think about some confounding variables. And again, a confounding variable is just other things that may be at play that might explain an increase in pulse rate besides the caffeine. And so some confounding variables might be, for example, the student's weight or perhaps their activity level, how long they've been sitting to begin with. It could have to do with their metabolism. Their blood pressure may also be at play. Whether or not they've been sitting the whole time, or maybe they were walking around after they drank the soda. You know, if they had to get up and go across the room to open the refrigerator to get the soda versus having the soda being handed to them while they're sitting. All those things might affect the pulse rate of the students 10 minutes after consuming the soda. This is another example where we started looking at the responses of individuals when they're being studied. In an experiment from the 1920s, 1924 through 1932, in fact, at the Hawthorne Works, which is a Western electric factory outside of Chicago, the company commissioned a study to see if its workers would become more productive in higher or lower levels of light. The workers' productivity seemed to improve when the changes were made and slumped when the study concluded. So many of you are familiar with the placebo effect. Well, the Hawthorne effect is when this effect was first, I guess, discovered or first, accredited. And I'm going to pull up the website. So the Hawthorne effect is basically the natural tendency of humans to react when they're being observed. So, and especially like in a situation where you're looking at worker productivity, well, any change in the environment, if the workers know that they're being studied, may increase their productivity. For example, some of you may have noticed that I can't remember if I did this in period 4 or not, but I pulled out a camera for some of my students and I left it on a tripod. Having the camera in the classroom may have actually changed the behavior of some of the students in class, may have made them more alert, maybe increased their level of engagement in the class versus if the camera wasn't there. So a lot of times people change their behavior when they know they're being observed. And then when they're not being observed, the behavior returns to normal. And this was first discovered like in the 1920s and 30s, and again, it's called the Hawthorne Effect. We call it the placebo effect, and again, it's the tendency of humans to show a response when they know they're being treated. And if you think about this electric factory outside of Chicago, if you think about the other variables that may have impacted the workers' responses, certainly if they've got a bunch of researchers taking notes on their productivity, that may have changed their productivity as well. All right, so we're going to talk about ways to experiment well. We did this with sampling designs. Remember, the whole goal is to reduce bias and make sure that the data that we collect is worthy of further investigation. And so one of the ways to remedy confounding is to perform a comparative experiment in which some units receive one treatment and similar units receive another. Most well-designed experiments compare 2 or more treatments. Comparison alone isn't enough. If the treatments are given to groups that differ greatly, then we're going to have bias. The solution to the problem of bias is random assignment. And again, we saw this with the samples. When we had selection bias, the way we avoid selection bias is to randomly select people for our samples. So the difference between sampling and experimenting is with sampling, we are randomly selecting people from the population. With an experiment, we're often dealing with volunteers. So we're not randomly choosing people from the population. We usually have a group of people that are willing to perform an experiment, and then what we have to do is use randomization to assign them to a different treatment group. So random assignment basically means that the experimental units are assigned to treatments using a chance process. There are essentially 4 elements or principles of experimental design. Comparison is one of them, but some textbooks actually lump comparison into control. So sometimes you'll just see that there are three aspects or three principles to experimental design. Primarily, random assignment is extremely important because it reduces bias. Using chance to assign experimental units to the treatments, it helps create roughly equivalent groups. Just like we wanted to have a representative sample from our population, we want the groups or our treatment groups in our population to be very, very similar. And so the way we keep them similar is by using random assignment. And that way it sort of balances the effects of the other variables among the treatment groups. We also need to control all those other variables. So you want to make sure that, if you're performing an experiment, that whatever you're doing to the treatment group, you're doing it in the same way to the control group. So you want to make sure that all of the effects are the same for both groups, and that the only difference between the groups is the treatment. So if you were doing an experiment on weight loss, for example, you wouldn't want your subjects to change their behavior in any other way other than taking the weight loss supplement. Replication is another important aspect of experimental design, and it can be interpreted in two different ways. Generally, replication means that you want to make sure that you're using enough subjects so that you're kind of reducing the variation that could be between your results. So use enough experimental units in each group so that any differences in the effects of the treatments can be distinguished from just the chance differences in the groups themselves. Replication can sometimes also mean that the experiment was conducted several times and the same results were found. But generally speaking, replication means you want to have a lot of subjects in your experiment. And a lot doesn't necessarily mean like 200. It could mean 5. But you do want to see the same results occurring for many individuals. And like I said before, comparison isn't always considered one of the principles of experimental design because it's lumped into control. If you have a control group, then it implies that you're comparing a control group to a treatment group. So the comparison is already implied within the control. So what I'd like to do now is have you guys watch a short video. This is another one from the Against All Odds series. And then when you're done, there are some problems out of the book. You can either use class time to complete the problems out of the book. The textbooks are going to be located on the shelf behind the whiteboard that Ms. Davis uses. Or you can work with your partner on your project. And what I want you to do at this point is start looking at the survey itself. Like, first of all, now that you know what your population is, if you would like, you can go ahead and label your population and select your sample. You're going to be doing this randomly, and you must do a simple random sample, and you must explain in words how you determined your sample. And then you also need to construct your survey. And I am going to want to see that before break. So I'm asking to see that, I guess, Monday or Tuesday of next week, if you could have your survey ready, because I would like for you to have all your data gathered before Thanksgiving break so you can work on it over Thanksgiving. But for now, I'm going to go ahead and we're going to watch a short video on Against All Odds.
Funding for this program